Welcome back with the end of chapter 19, and what I want to look at here is the neat technique which lets you look to see where proteins are localized in cells, which obviously be useful because where they are gives you some information about the function, and how you can do that using fluorescent protein tags. And so we're talking mainly about green fluorescent protein, but there is red fluorescent protein and yellow fluorescent protein, and some variations on this stuff. Originally discovered and employed by a man called Roger Tien, and he won the Nobel Prize for that. So it's really interesting stuff. Let's start off pretty general. So we're looking at here the localization of a protein in the mitochondria of some different cell types here. And that mitochondrial protein has been genetically modified so that one end of the protein has fluorescent protein added to it. So what you have is you have the protein of interest, whatever it would be, that localizes in the mitochondria, and then on the end of it you've stuck a yellow fluorescent protein. So this EYFP stands for enhanced yellow fluorescent protein. So you have a fusion between the protein that you're curious or interested about and a yellow fluorescent protein. Is what you hope is that when you make these fusions between this gene or protein of interest and your fluorescent protein, is that when they both fold up, neither of them interferes with the normal localization and function of the cell. So if you take a mitochondrial protein and you tag it with an enhanced yellow fluorescent protein, so you make one of these fusions, and then you introduce that protein, or is what you actually do is introduce the gene that encodes your protein of interest, here it's the mitochondrial protein and fluorescent protein, into cells, you can use fluorescent microscopy to see where the fluorescence goes. And so you can see all these different kind of patterns of mitochondria that you get in different cell types. And then you can see that, what's neat about this is that mitochondria aren't at all the shapes you see of these nice kind of oval structures that you see in textbooks. They can be long, thin, threaded, in some cases branch structures. So you could even learn something about the structure of organelles by sending a protein to an organelle and looking to see what the shape of the organelle is. So that's the kind of data you can get from using fluorescently tagged proteins. Let's look to see how you actually do this. And I'm not going to give too much detail on this because this is kind of, again, this is a little bit of genetics, but it's a genetic technique used by cell biologists. So we could take we could do some genetic trickery, which we'll look at over here on the left, and we could create a C. elegans, which is a type of little nematode, which is expressing a fluorescent protein. Then we could see where it is in the worm, and this actually looks like it's in the gastrointestinal tract, whatever this protein would be. So we could look at the localization of claudins or occludins or cadherins or integrins or any kind of protein, nucleolamins, any kind of protein we wanted by adding on a fluorescent protein tag. So, excuse me. How do we do this? Well, here's the gene structure. So this should kind of be relatively familiar to you. So we've got a promoter region, and so this is all in DNA. We've got a promoter region, and there's a 5' UTR, and then we've got a part of exon 1, an intron that will be removed, an exon, an intron, and another exon. Now what we can do is we can genetically engineer this gene so that we put GFP at the end of the last exon, and so after the GFP comes the stop codon, and there's a 3' UTR. Now if you introduce that using some genetic tricks into the C. elegans genome, is what happens is when you get transcription and translation, the introns get brought together along with this last piece of exon 3 here, which gives you the GFP tag. So what we have is the protein we're interested here tagged with GFP. So when this gets translated, we end up with the protein of interest with GFP just literally stuck on the end of it. You can do this at the N-terminus, so that's a C-terminal GFP tag. You can do it at the N-terminus, where you put the GFP at the beginning of exon 1, and you get, instead of the GFP at the C-terminus, you get it at the N-terminus. So if you can introduce this piece of genetic material into the worm, then when the worm expresses this protein, then you can see where the protein goes. And of course, you're hoping that the GFP doesn't interfere with protein structure or protein function or protein localization, for example. So you can do this at the level of the genomic DNA, modify the genomic DNA. That's pretty challenging, but you can see then the fluorescent protein in in the worm. And as well, I'll do in the zaption is I'll add some other animals which have been made fluorescent. So there's fluorescent monkeys and fluorescent mice and all kinds of things. And we're not going to get into the ethics of doing that, but you can do it. The other way you can do this is you can introduce your gene of interest in the form of a complementary cDNA, a complementary DNA. So if you take a piece of mRNA that's been fully processed, so that's all the exons joined together, and you use an enzyme called reverse transcriptase, you can make a piece of DNA from the RNA. Then you take that piece of DNA and you put it into one of these circular pieces of DNA called a plasmid, and what we can do is we can put the cDNA here. And the cDNA is an open reading frame, so that means that from the start codon to the stop codon is all one set of 3 codons being read out together. So that's called an open reading frame, and genetics will help you with that stuff if you're not taking genetics. And so you can put the cDNA in here, and then you have the GFP. And so here we would have the cDNA producing our protein of interest directly linked to the GFP. And then this sequence here adds a poly-A tail to the mRNA that would be produced. And then all these other sequences here are to get this piece of DNA expressed in eukaryotic cells. So we could take this piece of DNA with our cDNA for our gene of interest, and we could put it into, for example, some cells which are being cultured in vitro in dishes, and we could then use those cells to look to see where does this protein go. So don't worry too much about the genetics here, but this is a much easier way of doing this experiment compared to this way which we just looked at. The take-home message here is that you can take a gene that you're interested in, and you can genetically manipulate that gene so you can add on a fluorescent protein to the end of the genes. When the protein's made, it's a fusion or a hybrid between your gene of interest and a fluorescent protein. And then when you introduce that genetic construct into cells or an animal, you can look to see where the fluorescent protein goes and therefore learn something about the relationship between localization and function. So that's all we're going to talk about with that technique. That's it for chapter 19, and I'll see you soon in the next set of material.